Okay, welcome to the fourth lecture on biological signaling 2. And you can see the overall title is the TRPV6 protein. That is the one protein, one ion channel from the TRP superfamily. We will have a look at this superfamily in a second. Is the TRPV6 protein a subunit of the calcium release activated channel, that is the crack channel in mast cells. And let's go a little bit deeper first in the process of allergy, because that is how it's particularly linked to, and fresh up some of the points you already may have heard or you know from allergy. So there are many possibilities for our body to recognize some, let me say, compound as an allergen. There is only one compound where it's clear that our body will not recognize it as dangerous, not as an allergen, and that is sugar. Because sugar is particularly needed for our brain as nutrition, as energy source level. But that's the only exception. Against any other type, as I have read in literature, our body can, our immune system can recognize as dangerous and can develop allergens. So what are the most prominent ones? Okay, we already discussed this in biological, in the practical 2 course, biophysics practical 2 course, for instance, pollen, which are just now, hopefully when it gets warmer, maybe not these days or the last week, but we will hopefully get warmer soon in spring, that the pollen from various plants are flying around and are recognized from our immune system as dangerous. So these are potential allergens. Very prominent, I already mentioned, is injected materials, injected compounds that can come usually from insects, insect venoms, for instance, from bees or from wasps. But it could also lead, it's already just right now a matter of discussion against vaccines. Because you know there have been some problems with, let me say, problems under a certain remark, problems with the COVID-19 vaccination with one particularly vaccine, that of AstraZeneca. But what here occurs is a very rare side effect, although it can be severe. There have been indeed some deaths. It seems that it's not an allergic reaction, because usually these allergic reactions occur quite rapidly, particularly these type 1 immediate allergic reactions. They recur within 50 minutes. So that's the reason why when you get vaccinated with these new anti-COVID-19 vaccines, usually you have to stay for about 15 minutes to see whether there is allergic reactions. And it turned out that the classical allergic reactions, meaning you have some drop in blood pressure, which could really be dangerous, are very, very rare. But there have been this side effect, and I've just recently talked to a new professor of pharmacology from the medical faculty. It could indeed be that very rarely this vaccine or the antibodies that are generated by these vaccines in some persons could lead to an activation of thrombocytes in a way that thrombocytes get reduced in our body, and at the same time thrombocytes, these are blood platelets, the thrombocytes, that they somehow lead to some thrombosis of these in various types of venous systems, particularly in the brain. This, of course, can be dangerous, but meanwhile, it's sorted out how it can be treated by, when it's identified, it can be treated by a high dose of specialized IgG injections. But you have to somehow realize that this is somehow linked to the vaccination. But let me just say that it's 1 in a million where this occurs with this, particularly with this new vaccine of AstraZeneca. And maybe the reason why it has not been found with the other vaccines from Pfizer, for instance, maybe that they have used it particularly for elder persons, and it seems that in elder persons this develops. This usually will not develop this thrombosis, but it develops with a higher rate in females than in men, and particularly in females in the age between 30 to 60, something like this. And maybe one of the points why this has led to some numbers actually where this has been identified is because when in Europe, not in Great Britain, but in Europe, here we started to vaccine with AstraZeneca. It was at the beginning not used for elder people because there were no clear studies about it, but it was particularly used for younger people, for females in the hospitals, in health services. And maybe this was the reason why here some of these critical side effects occurred. But it might be linked to the application to this certain group, and the Pfizer vaccine, at least in Europe, is only, or is most of the time applied, or has been applied only to elder people. And in elder people, it seems to usually not to occur. So yeah, that's some of the side effects, but that is not an allergic reaction. But I just wanted to mention because also vaccines are mentioned here. Drugs are also well known. You can develop very typically, for instance, a drug, an antibiotic, penicillin. Sometimes it can happen that even in a hospital where you're working with penicillin, that even a very small dose of penicillin in the air, because it has been applied to a patient, can even lead here to quite a dramatic reaction in terms of an anaphylactic shock, which is a very strong anti-allergic, very strong allergic reaction of your body because you are allergic against, you have developed an allergy against penicillin, for instance. There are other ingested materials, like, for instance, food, just nuts have some high incidence of developed allergens. For instance, peanuts that were quite regularly supplied, maybe it was some years ago, by airplanes. And in US, it has turned out that many of the people there, they develop allergic symptoms against peanuts. So peanuts on all US flights have been meanwhile forbidden for quite a long time. But in principle, against any stuff in our food, you can also develop allergies. And of course, you have contact allergies. Whenever you get in contact with a material, very classically is related to some jewelry, which contains nickel, that you get a nickel contact allergy in terms that you develop a red point. I do not know the English word, but for a Gürtel, when it's directly on your skin, and the Gürtelschnalle that contains nickel, but also jewelry, which is directly in contact with your skin, that can also generate contact allergies. But also from plants, some of them have a very high potential to evoke allergic reactions. So there are several types of allergic reactions. We particularly focus on the very fast type, the type 1, immediate hypersensitivity, as it's called, which occurs at the fastest within 5 minutes and goes to about 30 minutes. You can see there are other types, and some of them actually occur quite over a longer time frame. But one of the most critical is, particularly if it develops as a strong allergic signal within your body, is this type 1 immediate reaction, which can lead to anaphylaxis. So what's going on here? And first of all, classically, one of these allergens are nowadays pollen. Pollen are inhaled, you know already from the practical course of this handout. Pollen are inhaled. They are taken up via our bronchial vessels, and they are digested. They are taken up by these antigen presenting cells. They are taken up, they are digested, then they are put onto the surface, and then they are recognized by T cells, and particularly also T helper cells. And then there is a connex between these T helper cells, you can see here, that gets activated, in as far that the T helper cells help to activate the second branch of our immune system, the B lymphocytes, the B cells. And the B lymphocytes, when they get activated in contact with these T helper cells, then generally the B lymphocytes are those cells that finally lead up to the secretion of antibodies. And one particularly type, which is in a high concentration or ends up in a much higher concentrations in patients that show allergic symptoms, are these antibodies of a class. Another word for antibody is immunoglobulin. And you will see later on, we have 5 classes of immunoglobulins. And this class, which is in a higher concentration in the blood, is the immunoglobulin E. So antibody of the E class, the IgE, that occurs in our blood at a higher concentration. So this occurs at first contact. And whenever you have a first contact with an allergen or generally an antigen, nothing will happen. But with an allergen, after the first contact, you can say, okay, but it's fine. An antibody is developed to bind an allergen, and by binding it is somehow secreted from our body. But there comes another cell type into play, and this cell type is the mast cell, and you can see it here. You can also see it here. This is the plasma cell, the IgE secreting B lymphocyte that is secreted IgE, and the IgE, by floating around in our blood, it also finds the mast cells. And the mast cells, they have specific receptors for binding the IgE. And you can see here already IgE is bound to these specific receptors, and now it waits there. And it can wait there on the mast cells for several months. The point is, when you have now a second contact with your allergen, then a certain reaction starts which cannot easily stop, and we will come to that in a second. But let me just put your attention to what we have in a mast cell. The mast cells, on the one hand, they play a very prominent role in our innate, in our broad immunity, but in connection with the IgE, they get some very important role linked to these generating allergic symptoms. Within the mast cell, we have granules. These are these vesicles. And these vesicles, they are full of mediators that generate the allergic symptoms. We will come to that in a second. So here you can see how actually the receptor is called. The receptor for IgE, where actually the plasma membrane is full of mast cells, is called the Fc epsilon receptor type 1, because type 1, that is the immediate type hypersensitivity that leads to these allergic reactions within a time frame starting at about 5 minutes. So you see the receptor as a cartoon is composed of several subunits. The alpha subunit is the most important one. It contains the binding site for IgE. And you can see here, IgE is waiting for a second contact with allergen. And then something happens. What you can see here in the upper part, the antigen, it binds to 2 IgE molecules on 2 receptors at least. And by this binding to 2, it leads to a cross-linking of the receptors. And whenever these receptors get cross-linked, dimers are formed. And we will see this later. This generates a signal cascade that will result in the following. Here you can see that is a mast cell here, where actually with a certain dye, a red dye, particularly the granules had been made visible in a microscopic preparat here. Here you can see, we will see this, image later on as well, but here you can see an electron microscopy preparation. What happens here when you have a normal mast cell and after IgE is waiting on the mast cell, this mast cell is incubated then or gets a second contact with allergen. What happens within 5 seconds and 60 seconds? What you can very nicely see here, protruding out of the plasma membrane, you see these vesicles. They get more and more out of the plasma membrane. They fuse in an exocytotic process, and you can see it here. This mast cell is already full of these granules, so the content of the granules here has been released into the extracellular medium. And that can occur, as you can see here, even in a time frame of 60 seconds, and then it takes some more minutes before symptoms actually will develop. You can see it here as well in two opposing or parallel images. One that is the mast cell where these granules have been dyed. Here you see the electron microscopy image where you have this black contrast, each of these black dots here or holes that actually represents a vesicle, a granule. And after 5 minutes of activating the mast cells with IgE plus allergen, you can see what is happening here. You can see the red dye is almost completely lost out of the cytosol because it's released to the extracellular solution. So here it gets quite diluted, so it's not visible anymore. And particularly here you can see also the filled, here still filled granules, they already have lost their filling, so they have not this dark contrast. And you can even see here maybe some of these granules, they are in the few, they just have in this process of fuse to the plasma membrane and release their content. So the point is, that's the point of no return. Then the granules release their content, and in addition, several reactions are turned on within a mast cell. So what are particularly mediators that are released here in a very fast reaction? And one of the prominent mediators, I've already mentioned this due to these antigen cross-linked IgE Fc epsilon receptors. One of the prominent mediators you may have heard of is histamine. Histamine and also some lipid mediators, that can occur. Histamine is the most fastest one because it's already stored together with some enzymes in these granules, but there are also some lipid mediators generated. That takes a little bit longer, but they lead to vascular smooth muscle response and to immediate reactions. We have later on reactions where additionally mast cell also some of these transmitters of the immune system, or let me say cytokines or interleukins, we will see them later, are generated. They somehow belong to a longer late phase of reaction where you can see inflammatory signals. We can see this also in a second, how it looks like. So this is the immediate reaction. This is a late phase reaction. So immediately, for instance, when you have been bitten by a wasp or a bee, so typically you have a sort of allergic reaction. You can see here is where the bite has occurred. You can already see one of these signs of inflammation, that your skin gets red. In addition, it gets swollen. Swollen is because you have some edema where some fluid, particularly water, has been released from your blood vessels because your blood vessels will get leaky due to histamine. And usually, I guess maybe almost everybody has already been bitten by a wasp or a bee. So usually, okay, it hurts to some extent, but usually these signs, they will disappear usually within 2 to 3 days. But when you have some, let me say, proposition that you have a certain chance to develop an allergic reaction, even a stronger one, when you still have these signs of red skin, now you see at the larger area, particularly within 1 week. So you have to take care, because it's not predictable at the second bite, for instance, by a wasp or a bee, how strong your reaction will be. If this really goes on and stays longer than for some days, so it goes on for 1 week or 2 weeks and gets even larger, then you might have a preposition to develop really an allergic reaction. The problem is you cannot foresee what happens, but there are tests around that allow from your blood to see whether you actually might be, and then let me put it in this way, you might be a patient that could actually have a quite strong allergic reactions, because when it's only locally, okay, over the arm, there's no problem. But when it's a reaction in the whole body, meaning particularly in your arterial blood vessels, then you can have problems. And I will come to that in the course of this lecture in the next minutes. So what actually is released? I've just mentioned before these toxic mediators, and the most prominent one is histamine, which makes particularly the blood vessels leaky, increase vascular permeability. So you always have water flowing out from your blood vessels. And I will come to the symptoms, whether they are local or in a more generalized form, also in the next slide, but also enzymes. And you see here, these enzymes are classically proteases, so they destroy proteins quite unspecifically. So they have also some remodeling of a connective tissue matrix. These are the cytokines, interleukins already mentioned. What also is mentioned here, these are lipid mediators. We will see how they are generated, particularly belonging to the class of leukotrienes. Because these leukotrienes, they have been, or they might play a certain role with smooth muscles, and particularly you see a mucus secretion with the smooth muscles in our lungs. So I will come to this on that slide here. So as we've heard, histamines or lipid mediators, they make, particularly histamine, make the blood vessels leaky. If this is controlled only locally, for instance, occurring in our nose, then you have a running nose because water gets out of your blood vessels. That at least makes a running nose, this rhinitis. When you have eyes with tears, also as an allergic response, it's also locally. But when you can imagine that when you get a bee or wasp venom injected, it enters your blood circulation. And when you have a strong response now in your blood circulation, then you can see that these leaky blood vessels, that looks like for our system, for our circulation, as a loss of blood. A loss of blood in terms of a loss of water flowing out of the blood vessel leads to a drop in the blood pressure. And when the blood pressure drops, not only locally, but also in our arterial system, then you can really have problems with a strong effect, because this looks like a shock symptomatic. And this is also called the anaphylactic shock. And I will talk a little bit more in the next slides. If it's only locally in the tears in the eyes or in the nose. Okay, it's not, of course, comfortable, but everybody can live with that. It will get more dangerous if it involves the lungs, because in the lungs, one of the reactions to the histamine, and particularly also to the lipid mediators, is that our lung vessels, also composed of smooth muscles, they will contract. And the contraction of the bronchial vessels in our lung, that interferes with the breathing. And the bronchoconstriction, another word for that, is asthma. So you can have an asthmatic attack, particularly when you are experiencing these allergic symptoms for a longer time frame, meaning for months, for instance. So whenever you have an involvement of a lung, you have to take care, because when it comes again every year, usually the reactions of your lung will get stronger and stronger. And it's well known that you have some rebuilding in your lungs going on to this, due to this inflammatory process. And this can really lead to quite severe asthmatic responses. Particularly with children, they sometimes can be easily tested whether there is an involvement of a lung as well. There can be the respiratory volume can be controlled. Whenever you have a bronchoconstriction, whenever you breathe in, you cannot take in as volume as it would be expected. Interestingly, children, they do not feel it quite early. They can have an a reduction in the volume by about 50%, and then they start to feel it by don't feeling so well and getting not enough air. But they have a high reserve here that only can be determined by a pediatrician, a medical doctor for children. And when you do see these during periods where, for instance, children have allergic symptoms, there is a need to treat this. And particularly, there are two drugs which are usually also even combined. One is a drug that relaxes, that makes the the bronchial vessels wider again. This is one drug we already have heard, salbutamol. And the second drug, that is, it can be cortisone or cortisol. That is a subtype or a further development of cortisone. Which are both drugs, let me say, they can be easily applied because you don't have to swallow some pills, you have to inhale it. After a certain training at the beginning, it's quite easy also for children. And the second drug is butenoside. Butenoside, it's just in the newspapers, just the last few days, because that's nothing else what is regularly used to treat or to prevent asthmatic development for persons who have allergic reactions in the lung. It prevents inflammatory processes. And it turns out, although studies are currently under review, that this drug taken at the right time when you have a COVID-19 infection, particularly when you are at the stage where your oxygen level in the blood drops under a certain level, meaning under about 90% or something like this. This is the stage where it can still be decided whether you have to go in a hospital and maybe you need to be treated by intensive care, or, and it seems that this drug, by taking at this time point, it can simply by inhalation, it can help to prevent these strong inflammatory processes and to prevent the development of these really critical reaction in the lungs due with COVID-19 infection. So that just came out, and interestingly, that is a drug which is also very regularly used. I would say with some people think cortisone has a number of side effects, but when it's only used in a very low concentration and only locally because it's inhaled and gets only to your lungs, then you can forget about strong side effects. But it's a very useful therapeutic drug. So whenever it gets involvement of the lungs, you have to take care, and that needs to be seen and treated by a medical doctor. There are other side effects. For instance, you can develop intestinal hypermotility in terms of getting a diarrhea. Also, inflammatory processes involve other tissues, which, as mentioned before, could lead to some tissue remodeling. And tissue remodeling, it takes some time to get reversible. So these are the effects that actually can be caused by this degranulation of mast cells. The strongest effect, as we already have heard, is the systemic anaphylaxis. Yeah, systemic anaphylaxis. You can see here, classically, it's caused by those where you have an entrance of the allergen into the blood circulation. That's needed. With peanuts, of course, it takes some time, but with venoms, when something is, for instance, gets into your serum, vaccination or anything else, or drugs, when, for instance, penicillin are applied, that can develop quite early. And we will see the time frame and what happens to the arterial blood pressure in a second. This is locally, but it must not stay locally, as already mentioned. But insect bites, okay, if it stays locally and is gone within some days, you do not need to go further for an allergy testing. Pollens, okay, they are, of course, not very nice to inhale and to get these allergic reactions, but usually the allergic reactions occur to a certain area, for instance, to the nose or to the eyes in terms of allergic rhinitis. Asthma is more dangerous and needs to be controlled because when it occurs, and usually when you develop an allergic response, whenever your body has it, classically, it stays on from year to year. It can even get worse. Very rarely, it will by itself disappear. It could be, but usually it's not the case. And keep in mind, nowadays, about 30% of our population, they have these allergic reactions. And this percentage is increasing more and more. And keep also in mind, you can develop allergic reactions at any age. So even I'm lucky here, even I have no allergic reaction, I could develop it, let me say, in the next 10 years. It's possible. It depends on your body. And of course, food allergies, they can also get quite strong. Particularly, there are also cross allergies that similar types of allergies, for instance, are found in various foods. So whenever you have an allergy against one food, you also could have at the same time a cross allergy against another food. But that's quite well documented in more detailed publications. So here,
I have a question. So regarding histamine, if I am now histamine intolerant, is that an allergical reaction? Not, or
no.
But if I'm histamine
when you are now, because that's also a problem. I share this in part, this histamine intolerance. It's not due to an allergic reaction. The point is usually, particularly in the food, the elder it is, and depends also on the type of food, it can be that the food contains a high concentration of histamine, or that could also be, it helps that histamine is released in your body. But usually, let me concentrate on the food you take up and which is digested. Usually in our intestines, there is an enzyme available that is particularly well prepared. It's called histamine oxidase to destroy the histamine. It turns out that people who suffer from these histamine intolerance, they, due to some development, that they miss this enzyme, or the enzyme gets less and less. There are several types to treat these. The one is, for instance, this enzyme can be taken up just before you go for lunch or for dinner, and you have problems with the some of these food you may like to take up. For instance, tomatoes are one of those, or what is very prominent, but that I guess will not occur every day. For instance, champagne. Champagne has one of the highest levels of histamines. And I had one, let me say, one very impressive experience here, because a colleague at the institute, she got some grant funded. So that is always a matter of celebration. And she had access to a very nice champagne. And here I took 3 glasses. So usually I do not drink that much, but 3 glasses because it really tasted very good. But afterwards, because it's known you can control, and it's in many publications, it's documented how high the histamine content is. I didn't know it before, but champagne had one of the highest histamine contents. And what I developed afterwards, I developed a sort of, let me say, my head gets quite red and hot, and what is, I would say, not very comfortable, an increase in the blood rate. So in the heart frequency rate. So I had a frequency from 60 to about 100, and I think this also was related that I get an arrhythmic heart contraction. So your heart usually has a certain rhythm, but this gets arrhythmic, and that was bam 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 bam, and you can imagine when you have it, you really feel it. And I had this for 2 days. So that is when I got more into contact with what is related to this histamine response. And it seems that I also have these problems as I cannot, or my body will not tolerate high histamine levels. And the second was because usually when food is stored, for instance, red wine has higher levels than white wine, but there are meanwhile wines produced that contain almost no histamine. But what also you have to take care for instance, fish, whenever food with a high concentration of proteins like fish or like mushrooms, for instance, when they get older, they liberate histamine they liberate histamine. And for some people, there is no problems. But that was a second, I would say, really not very nice experiments. We all had together during a Studienkommissionssitzung. I mentioned to you already in Attersee, we had a Lachsforelle, a trout, a salmon trout. And it seems maybe it was not completely fresh, but no other complained. I also did not complain, but about half hour later, I almost lost my consciousness. So I had to stay for about half an hour, sitting in the car, and whenever you feeling getting unconscious, okay, that was my experience, and that was my second point, where I think that was confirmed. I cannot tolerate high histamine levels. But whenever you know it, you have to take care and you have to test it individually, what you can tolerate or not. And finally, you will learn to work with that.
Okay. Is then the combination of allergic reactions and histamine intolerance then more dangerous, because on an allergic reaction we produce more histamine, and if I'm histamine intolerant, then I can't compensate this.
Yeah, but the one is particularly the histamine which is released or which you have developed by food, particularly in your intestine, and which is then taken up. Yeah. The other is that is maybe only locally released in your body. But of course, if you have, let me say, from my case now, just drank a lot of champagne, and okay, I'm lucky here, but when I would have also an allergic reaction against bee venom, and at the same time bitten by a bee, okay, that could lead to quite high levels of histamine. But what helps is to take up this enzyme, this histamine oxidase, but that you usually do not regularly. When you know, okay, you will have a nice meal, maybe with drinking some wine and taking up some, for instance, also, tomatoes or Parmigiano, Parmesan, that could also have high levels, contain high levels of histamine. What I usually do to prevent this, I take an antihistaminicum. We will come to that in a second. This helps also, yeah. It binds to the histamine receptor and prevents binding of the high doses of histamine you have in your body, and that lasts for almost a day. So this helps also, but you usually do not use it regularly. Okay, maybe this answered some of you, or also maybe some questions of the other students, because it's not so rare, meanwhile, that people have an histamine reacting body, and particularly due to several foods they do not well tolerate.
I have one question. If you have a histamine intolerance, can you develop allergies just as easily?
No, that's a different process. No, that's a different process. I haven't heard that this is really connected. No.
Oh, okay.
I don't think so. I don't think so. Okay, just talking about food allergens, but that's something different because this involves usually the mast cells, but as we're just talking about histamine intolerant, this particularly involves the histamine, which is contained in our food we take up. So this is the dramatic response in terms of an allergic person who is bitten by a wasp, let me say, and who knows or maybe did not know that he or she will develop an allergic response. You can see here, this is the regular blood pressure, typically in the arterial blood system, usually at the level of 100. So whenever this bite occurs and the venom is taken up, maybe it takes some time that it develops because you need to get it in your blood circulation. But after it is in your blood circulation, you have this very dramatic response. And you can see here your blood pressure now here from 80, which is typically quite low, drops to a level of about 40. So at the level of 40, that's our millimeter mercury. So at the level of 40, you are in a shock symptomatic, and you can be pretty sure when it drops even more, that's a problem for your body to still focus with the blood that is remaining, because the blood vessels get leaky. Most of the volume is now, or not most, but some volume is now in the extracellular medium and not in your blood vessels anymore. That's the reason why you have this drop in the blood level, in the blood pressure. At this level, your most important organ are at the limit to get enough blood. So whenever this drops maybe even more, or even this is a point where it can be dangerous, then your heart or your particularly your kidneys and all the inner organs, they cannot be supplied with enough oxygen. And this can lead, and you can hear it every year, that somebody who did not know, or maybe persons who know it, they have these very strong allergic symptoms. They do not have an antidote, meaning an emergency set with them to take care of that, and then it can happen if nobody is at the side with them, that they develop shock symptomatics that leads within 5 to 10 minutes to death. That occurs, unfortunately. I would say we have 5 to 10 persons every year. So what's shown here is when you know you are in danger to develop such a strong allergic reaction, you have to have this emergency set always with you, particularly when you use, that can happen in nature during a hiking tour, getting bitten by a wasp or a bee, that you have such a reaction. When you are getting bitten, you take out the syringe and you inject a high dose, what usually is used here. Here it's called epinephrine. Usually it's in German, it's noradrenaline. That is something that whenever you digest it within a very short period, it leads to a recontraction of your arterial blood vessels. And when the vessels get contracted again, this of course leads to an increase in the blood pressure. And that is what you can see here. By injecting this epinephrine here, the blood pressure gets back again. And this is the injection of epinephrine, which is shown here, how it increases in the blood plasma concentration. So this is directly related to a re-establishment of the blood pressure. And at a blood pressure of about 80 millimeter mercury, you can of course survive. But when it stays at that level for a longer time, yeah, then you are very close, or maybe you can die. So this emergency set that is available, it's typically prescribed by your medical doctor, but it's available in all pharmacies. And the main point is, take it with you when you know you can have such a strong reaction, because it will not help when you say, okay, it's at home, but I don't have it with me. What can help a little bit with these allergic reactions is, for instance, taking these antihistaminicum drugs. And also what helps somehow to counteract the allergic symptoms is to take a high dose of this cortisone or cortisol, or it's sometimes called also prednisolone, that is a derivative. But it takes some time, because usually you have to you have to swallow those pills. So that takes some time. And here you don't have time. You need this injection of epinephrine immediately. Otherwise, you will not survive. So just to point out that allergy can also lead to death by this very strong reaction, which is called anaphylactic shock. So particularly, that is just mentioned here, atopic asthma, as I already mentioned, that can develop with the lung vessels, with the bronchus, which you can see here. It seems that mast cells around our bronchial vessels, when degranulation occurs here due to the cross-linking of the Fc epsilon receptor, it leads to a constriction, as we already mentioned, and it leads also to an increased production of mucus. So both make the breathing more difficult, and that, as we just heard before, leads to this asthmatic attack, or to this asthmatic reaction, and needs also to be treated. So what are the main players now here involved? I already mentioned this antibody, or this immunoglobulin, IgE. Then, of course, the allergen. Then there are 2 or 3 cells that can, all of them can take part in that reaction, because all of them, they have the Fc epsilon receptor, and all of them have granules that can be released, their contents, and one of the contents of all these cells is histamine. They are classical type mast cells, but also basophils or eosinophils. They also can take part in that reaction. And whenever you get a blood determination, looking at whether you are a person, maybe you want to check whether you could be an allergic reaction person. Usually the immune cells will be analyzed, and particularly here the eosinophils. So a high concentration of eosinophils is one indicator that maybe you can have an allergic reaction just going on. It's not all, but it can be related to that. And the second is, when you have a close examination of the immunoglobulins, here it's also very prominent. You will get a higher concentration of IgE, and that is a further indication that an allergic reaction is going on in your body. And then you have to identify what is the allergen. And based on this, maybe you can go on with more specific treatments. We already heard of the mediators, just like the histamine and lipid mediators, but also interleukins. So let's have a closer look on IgE. Usually the IgE, the antibody, the immunoglobulin, is responsible for this type 1 immediate hypersensitivity. As referred, within 5 minutes, you can have strong reactions. Although usually it's a very small amount we only have of IgE in our body, 0.001% of the all circulating IgEs, immunoglobulins, but there will be an increase when you have an allergic reaction. Just to mention, we have on the whole 5 IgEs, 5 immunoglobulins. IgG, that is the most prominent one, which takes care of a long immunity against, for instance, measles, when you had developed it or when you get a vaccination. Then we have IgM, which usually occurs during an infection in the first 1 or 2 weeks. But then it will drop again, and then the IgG level, they increase, and they can stay on, but they must not. They need not to stay on for a longer time, but sometimes, for instance, whenever you had measles as a child, usually the IgG antibodies against these measles, they will stay on for usually a lifelong time. We'll see it in a second. The antibody is like an epsilon form. So the Fc, the constant part for all IgEs, will bind to the Fc epsilon receptor on mast cells. You can see it here. So this is the constant part for each type of immunoglobulin. So that is the Fc part. That is the part that binds to the Fc epsilon receptor. And these are the variant part. That is the Fab, the antigen binding parts that are in parallel. 2 we have on each immunoglobulin. And via these 2s, you get from one to the next the cross-linking of the IgE, which is sitting on the Fc epsilon receptor by the allergens. Usually, half-life of IgE is only 2 days, but whenever it is bound, versus, for instance, 21 days for IgG, but for some various types, it can stay even longer. But you have to take also in account, we have also memory cells that always are in the body, and due to infection, they proliferate again and produce very rapidly IgG again. But the most important point, IgE, whenever it's bound to the mast cells, it can stay on there and wait now for, as I already mentioned, the second contact with allergen. So what happens? We just have seen it, immediate type reactions within minutes, but also late phase reactions, which can take 6 to 8 hours, which are most of them in terms of inflammatory responses due to these lipid mediators, which belong to the class of leukotrienes or prostaglandins, but also some cytokines that also leads to inflammatory processes. Here you can see that one ion plays a prominent role here, and the ion is calcium, particularly for this fast release, because we will discuss this during the next lectures very extensively. The calcium that enters through the plasma membrane, through a certain channel, ion channel, the cell, leads to an increase in the cytoplasmic calcium concentration and to a sustained increase. So that lasts for a longer time in the minute time frame. And this increased cytoplasmic calcium concentration in turn then is important for driving the exocytotic process, for driving the fusion of the vesicles with the plasma membrane to release the content of these allergic reacting mediators. That occurs within minutes. On a slightly longer time scale, as you can see here, we have also a calcium influx, which turns on enzymes which play a role here in our phospholipid degradation. Just to mention this, from phospholipids, usually at the second position of this glycerol frame, we have arachidonic acid, which is released by an enzyme which is called phospholipase A2. Lipase that splits phospholipids at position A2. It helps in the middle because we have 3 positions at A2 to release arachidonic acid. And arachidonic acid can then enter 2 different enzyme pathways, one leading to leukotrienes and the other prostaglandins. Leukotrienes particularly can lead to bronchoconstriction, and prostaglandins generally lead to inflammation and also to feeling. It's also well known as a hurt mediator. So whenever something hurts, also prostaglandin, for instance, are released. But the immediate process here is the release by an increased cytoplasmic calcium concentration. So the influx of calcium plays a prominent role. And of course, this leads now more to the molecular processes that are involved to drive this influx of calcium. And before we come to that, let me just, that is already shown to some of you with the practicum Biophysics 2. What can you do to prevent allergic symptoms? Of course, you can prevent the contact with allergen. But as I mentioned, to be bitten by a wasp or a bee, that just happens. You cannot completely rule it out. So here I can only refer to the emergency set if you know that you are a strong reacting person. For instance, pollen, you can avoid going to the mountains or to the sea for 1 month, but usually you have to take 1 month of holidays. That is, not very realistic. The other process is called hyposensibilization. Here you somehow try to get, first of all, you have to identify the allergen which you are reacting against. And if it's available in a quite purified form, you can start under control of a medical doctor, classically at the beginning in a hospital, but then under a medical doctor close to you, to get increasing dose of your allergen injected into your body. It's classically not into the venous, not into the blood system, but it's just an intramuscular injection. So what you actually want is that with the increasing doses, you want to stimulate the immune system, but not at the level of IgE. You want to stimulate your immune system that after a treatment with increasing doses, after a period of 3 years, what is quite usually here, you want to have a high concentration besides of the IgE. You will not get rid of it, but you want to have, in addition, a high concentration of IgG. And IgG, when you have it at a high concentration, whenever you get into context now against in contact with the allergen you have been treated against during this hyposensibilization process, the IgG is at first first at the site where you have the entrance of the allergen. It binds the allergen before it gets to the IgE, which is bound on the mast cells. And by binding the IgG, by binding the allergen, it eliminates it. That is one theory how the hyposensibilization actually can cure you. There are a number of remarkable targets here with the mast cell, what is called stabilization, particularly as we've heard, this calcium influx is the most important part here to drive the fusion of the vesicles. So one of the most important targets here to somehow inhibit, prevent this calcium influx by very, let me say, selective drug. But as already mentioned, it's identified, meanwhile, which protein is behind. So that was the reason why a number of scientists have been pursuing what is the calcium influx channel in mast cells. Whenever you have it, you can try to develop drugs, but at the moment, there are some drugs in the clinical test, but it's not yet available for all the patients that might need it. Classically, allergic reactions are treated by taking up drugs against two compounds that are already mentioned. The one is against histamine, so these are antihistaminica, or against leukotrienes. These are called leukotriene antagonists. So there are, particularly for the first antihistaminica, really a high amount of drugs available. What they do classically, the histamine makes, for instance, the blood vessels leaky by binding to a histamine receptor. And the antihistaminica, they do the same. They bind to this histamine receptor. They are sitting there with a high affinity. They prevent the binding of histamine, which is released. But the main difference is they are sitting there on the receptor, but they are doing nothing. So they do not make, as a downstream signal, make your blood vessels leaky, which pathophysiologically occurs by histamine when it binds to the histamine receptor. So that's the main advantage. Usually, it has no side effects. But one of the most serious side effects was that the older antihistaminica usually makes a person very tired, but this is solved. So there are classically no side effects, and usually people have to take these antihistaminica over the time where actually they show these allergic symptoms. But keep in mind, it will only help in part for the strong reaction of the anaphylactic shock, because maybe you have some mild reduction, but it will be not enough to completely block this drop in the blood pressure. So here actually you have to take care with this emergency set. Anti-leukotriene antagonists, they are already provided, but mainly for children because the effects are not very strong, which helps, and they particularly help if in children allergic symptoms also can be already seen at the lung in terms of a reduced lung volume. So it's very clear that pharmaceutical companies, of course, are interested to develop drugs here that completely prevent the increase in the cytosolic calcium concentration, because this in turn completely prevents any release of the granules, but also any activation of the later phase of these lipid mediators, because you will not allow calcium to increase for a longer time within the cell. So that's the reason also why we have set here a certain focus in the practical course, but it will also be a focus here in this biological signaling 2 lecture which brings you, gets you more familiar with calcium signaling in cells. So one idea that also have come up is, but only, let me say, as a model in an animal experiments, to develop, it's also a sort of drug. It's a drug in terms of that it's an antibody. Some companies tried to develop an antibody which binds IgE. So it's an antibody which binds specific IgE. But it turned out finally, at least it never entered the clinical phase. Maybe there were some additional side effects here, or it was not specific enough, or no, or also not efficient enough. So the question is to understand what is behind this sustained calcium entry. What are the proteins behind to have a target where we can develop a drug against it? And here is again the process shown. This is a more general terms, but whenever this Fc epsilon receptor gets cross-linked, here you can see generally ligand, but this belongs to the class of the growth factor receptors. As already mentioned, in these type of the receptors, we have a dimerization of two receptors, and there is no G protein in between to activate this phospholipase C, because here actually a dimerization of the Fc epsilon receptor by getting cross-linked, that leads to an increase in the tyrosine kinase activity here at the cytosolic site. And tyrosine kinase means tyrosines from other proteins get phosphorylated by this tyrosine kinase activity. And here actually there is a certain isoform of phospholipase C, where when a tyrosine is phosphorylated here by an Fc epsilon, by an activated or by a dimerized Fc epsilon receptor, this tyrosine kinase phosphorylates phospholipase C, it gets activated, and then it works in a similar way as here the G protein coupled phospholipase C. It generates IP3 as a second messenger. Inositol trisphosphate binds to the IP3 receptor. It releases calcium out of the lumen of the ER, because the ER is a calcium storage organelle. But the calcium here, that is not enough for this fusion process. We need additional calcium coming from the outside through calcium channels in the plasma membrane. And it has been actually turned out that it's not the calcium that is released that activates a calcium channel in the plasma membrane. This was a little bit misleading, but at that time, that's 30 years ago, where these currents, where these channels have been identified in terms of electrical currents, it has been termed, due to the function, calcium release activated calcium channel, QAC channel. But it's not the calcium that is released that activates the channel, it's the drop in the calcium concentration in the lumen of the ER that generates a signal that has not been known for about 15 years, that generates a signal here, and this signal activates a calcium channel in the plasma membrane. So one of the main targets historically here was, and that was a Nature paper which has been published in 2000, what could be the protein behind this calcium channel in the plasma membrane. So you already heard a little bit about in biophysics practicum that it belongs to the TRP superfamily. That was just at that time in 2000, just a little bit before, a new class of ion channels have been emerged, and it's called transient receptor potential, because actually that is a channel, one of those channels occurs in the retina. And it seems that in rods or in cones, it leads to a transient change in the potential. So that's the reason why the name comes from, transient receptor potential. There are several classes. One class which has been proposed in the Nature paper, it belongs to the vanilloid type, which is called TRPV. You can see we have 1 till 6 subtypes, and TRPV6 has been proposed to be the protein behind the crack channel. In other words, before, because that have been cloned before, it has been before actually the TRP superfamily has been identified, and here it has been called CAT1, calcium transporter 1. So you will see both names, but CAT1 is identically, is equivalent to TRPV6. So as it's mentioned here. So what are similarities between the classical calcium channel you can measure, for instance, in mast cells, which are known to have this calcium influx channel because they are involved in these allergic reactions? And what are differences when you express the CAT1 channel in an expression system like HEK cells, HAK cells, human embryonic kidney cells? I will talk about them a little bit later, but let me say, whenever you have the idea that a protein is involved, particularly a protein which generates an ion channel, you can try to get the DNA behind this protein. And you know, whenever this DNA gets into the cell, from DNA to RNA to protein, then the cell makes this protein. And the HEK cells, usually they have almost no ion channels. So they are a classical model to use or to get them into contact so that they get these DNA of CAT1 included into the cytosol. And here it's enough when it comes into the cytosol, because there is also a machinery where it is then translated into a protein. So whenever you want to test a DNA behind a protein, you bring this DNA into a cell, the cell makes the protein, the cell in that case makes the CAT1 channel, and then you can check for electrical currents. In these HEK cells, actually where in control, there are no currents. But whenever CAT1 is taken up and translated into a protein, you will see very nice the CAT1 specific currents. And it turns out that the crack currents in RBL mast cells, that is a mast cell line, RBL means red basophilic leukemia cells. So actually that are cells isolated from a red. They are more closely related to basophilic. We have heard them before, basophils, but basophils behave quite similar to the classical mast cells. And leukemia means they are somehow fused to leukemia cells in terms that they are a sort of cancer cell, meaning that the cancer, this cell, they grow and grow as long as you want. But although they are more related to a cancer cell, they still have the characteristics of a classical phenotype before of the basophil of the mast cells. They have these crack channels, and the crack channels only get activated when the endoplasmic reticulum is depleted of calcium. So you can see here some remarks on the electrophysiological characteristics. They have a high selectivity for calcium. They have the typical current voltage relationship. As for many calcium channels, they can be blocked by lanthan, because lanthan with 3 positive charges is well known for blocking calcium selective channels. So both channels, the crack channel compared to the CAT1 channel expressed in HEK cells, all these have these characteristics, and they can permeate even sodium if you remove all the divalent cations. But that is maybe some speciality. What there is, the largest difference is that they show a different mode of activation. And let me just start here with that, first of all, methodology, and then at the first slide, where the difference is between CAT1 channel or TRPV6 expressed in a HEK cell and the endogenous, the inherently occurring crack channel of an RBL mast cell. You will see from now on, all experiments are in the whole cell configuration. So we typically see currents in the nanoampere, or let me say sub-nanoampere range, from a whole cell, from a whole mast cell, because there are many single crack channels overlapping, or many Cat1 channels overlapping. How are the stores depleted? Because we know the signal is only generated when calcium is released out of the stores. The first is a so-called passive store depletion. You know, with this technique, particularly when you see it in the movies, in the videos, which are supplied for the practical course, you can put a high concentration of a calcium chelator, that EGTA binds calcium with a high affinity in the glass pipette. And when with a glass pipette, when it's attached onto a cell, and you have a small hole that allows to enter the solution, the pipette, the interior, the cytoplasm of a cell, this solution also contains a high amount of EGTA. So whenever EGTA flows into the cell, it grabs any calcium. And usually the lumen, the ER, works in a way when calcium somehow leaks out from the ER, it's pumped back by an ATP. It gets pumped back by ATP into the lumen of the ER. And when there is no calcium now available, because all of the calcium in the cytoplasm is bound by EGTA, by the calcium chelator, then no calcium will pump back. So over time, what happens is you get a drop in the calcium concentration, because you always have a certain leak, and you have a pump that pumps the leaky calcium from the cytoplasm back. Ich muss euch anrufen, ich kann gerade noch nicht, gell? When it's pumped back, then it's fine. You have a high level of calcium in the ER. But when you prevent the calcium back, then the calcium concentration drops, and that is the signal to activate the calcium channels. You can do it actively. We have heard IP3 binds to the IP3 receptor channel, which is also a calcium channel. And IP3 releases calcium from the endoplasmic reticulum. That gets much more faster within, I would say, 20 seconds. This process with EGTA takes some minutes, but both lead to the same effect, activation of the crack channels. And just to get here an idea, and that is the final slide to show you, you will see from now on wholesale recordings where you can see here what is depicted here. This is on the x-axis, always the time. So you see seconds, it's a slow process. With increasing time of the experiment, you get an increase in the current. The current is shown as a negative current because that's simply conventionally done. A negative current that occurs due to an influx of calcium. And you can see here, at the beginning, we have established the whole cell configuration. Nothing happens. It takes some time for the EGTA to diffuse into the cell, some minutes in a range, or let me say, you can see here that seconds. So after 1 minute about, you can see a process which gradually leads to an increase in the calcium influx. Because when EGTA now binds the calcium, in turn, the stores get more and more depleted of calcium because nothing can be pumped back. And the more the stores get depleted, you can see the more this calcium influx current gets activated. And after about 150 seconds, you see here the maximum that is available from these basophilic mast cell-like behavior. This is the classical crack current. When, for instance, now that has been proposed that Cat1 or TRPV6 is the channel behind, when we now use DNA coding for the Cat1 channel, express it in these hex cells, one would expect something similar. But you see, there is one very contrary activation. Here it takes some time. The stores needs to be depleted. Here at the very first beginning, we already have here a maximum activated current. So CAT1, when it's expressed in hex cells, it's already active at the very first time. You see, even there is some gradual decrease, but it's well known that there are some calcium-dependent inactivations going on. But what looks pretty much the same is when you look here for the activated current here, it looks like this. You have here a point of cross-section in a range of about plus 40 millivolts. And you can see here for that current as well, you have also here, usually it should be in the range of about plus 40 millivolts. So it's also what's called the form is the inward rectification. So principally, the current voltage relationship look very similar, but what is completely distinct is the activation profile. Here in the mast cells, it takes some time, which nicely reflects the cracked channel behavior. The store needs to be depleted. This channel seems to be work independent of the stores. Although we also have stores in the hex cells, although these stores, they are certainly not depleted at the very beginning, the currents are already maximally turned on. And maybe one idea was maybe this protein would behave differently if it's in the same environment as the provided by the RBL mast cells. So the next idea, of course, was to express the CAT1 channel, not in the hex cells, but to express it in the same environment as where the endogenous cracked channel is already sitting. And meanwhile, it's well known that the environment, meaning the cellular environment, can affect functions of proteins. And that I will show you next lecture on next Wednesday. So I will stop now here. And thank you for your interest. We hear each other again next, not Wednesday, next Thursday.
I have some questions too.
Sure.
Can you also inherit allergies?
Sure. It's a disposition that when your parents, for instance, both have allergies, that at least you have a higher chance to develop allergies. It's not for sure, but you have a higher chance.
Okay. And for the asthma drug that could be useful for the COVID, what happens if you catch like a normal cold, if your immune reaction gets, I don't know, less?
I would not take it regularly because the cortisone is an immune suppressive reagent. Yeah, so usually it's fine that your immune system takes over. The problem with COVID-19 is that your immune system is somehow activated in to a very high extent that you have this extremely inflammatory process in your lungs. So it's really dependent whether you have coronavirus, but that can be tested. At least with these rapid tests, you have a result within 15 minutes. With these PCR tests, also within 1 day. So I would say that this needs first of all to be checked out before you switch or before you get prescribed this drug by a medical doctor. But your doctor, of course, should know it. But I would not take it only due to a suspicion. I might have it. That should be actually controlled. And that was pretty new in the newspapers right now. So I'm not sure whether it's still regularly used, but that needs to be clarified by a medical doctor.
Okay. And my last question, is there a reason why allergic reactions become more and more common?
It seems to be a multifactorial process and one which is behind because it particularly occurs in highly civilized Western cities, let me say, cities where people think the more cleaner our environment is, the better is it for our immune system. And that is wrong. because it's well known that that, for instance, children which grow up in a farmhouse in the countryside, they have usually a natural environment of a lot of viri and bacteria, and that's statistically significant. They develop less significantly allergies. But whenever everybody is completely clean, then you put an almost cleaning reagent made at 99% bacterial free, you have a very clean environment, and it seems that's how I explained your immune system gets bored. It needs something to do. And then it recognizes allergens, some stuff which usually is not dangerous to your body, as dangerous. And then that's at least one component how you can develop allergies.
Thank you.
Can I please ask you about silver fillings in the teeth? Can these cause metal allergy?
Which feelings?
Silver fillings. I know that in Austria they're not allowed, but in some other countries they are.
It's not silver. It's usually silver. We have gold. I know silver not. We have this which is called amalgam, which contains usually this mercury content to some extent. Yeah. Maybe they look like they have a silver color. Yeah, this is the amalgam fillings. Maybe you're referring to that, right?
Yeah.
Yeah. There is a lot of debate about that. It might be that some people that they can get some reactions with respect to this, but on the other hand, first of all, it's used for a long time period. Meanwhile, it's very well tolerated by many people, but there have been some, I would say, developments in generally that people, although they have no clear side effects, they get all these amalgam plums, or how it's called, fillings removed, and put instead gold fillings in, which of course are very, let me say, very expensive, or other stuff in it, these porcelain fillings. It's well known, at least I only can say I have these amalgam fillings in, and I have absolutely no problems. What my dentist told me, that it's well known, they still work for a very long time. So usually you can have amalgam fillings for 10 years. It gets a little bit more difficult with the other fillings. They do not have the same also specific behavior to, let me say, to temperature changes. For instance, sometimes when the holes or the caries in your teeth is pretty deep, I have heard that gold fillings, which are of course one standard because they are very inert, that you have when it's very deep, that the gold of course has a different capacity for reacting to change in the temperature. So whenever you get in contact to warm or cold, gold makes it much more sensitive, your teeth, to these differences in temperatures. For instance, these amalgam fillings, they are pretty similar to the original stuff. So it's a matter of debate. If you have no side effects, at least from my side, I did not change any of my fillings in the teeth so far. Yeah.
I also read the statistics of it, that women have more, like, higher allergy for metal than men. So maybe,
but
maybe it's worth testing for. I don't know. Is there like an uninvasive metal allergy test?
of course, you can do this. I'm not sure whether you have metal tests for mercury, which is some content of these amalgam fillings. But of course, there are allergy tests where I think about 30 to 40 probes are applied to your skin. And maybe one of these probes, at least one metal, which is very known to have a high allergic risk, is nickel. Yeah. With nickel, I assume that can be tested. You can even see it if you get usually with some nickel necklace or something like this. If you get red points, which somehow also you can where you have to scrabble there. That is at least some indication. And one point to see whether it comes to simple stop wearing those items which might contain nickel. Mercury, I haven't heard so, I have to say. But that's a matter of debate. But yeah.
Thank you very much.
Okay. If there are no more questions, I stop now and we see each other again next Thursday. Bye.
Oh, sorry, Professor.
Yeah.
Okay. I just sent you an email this week regarding practical training. So just when you have time to check it out.
Yeah,
it's about possible supervision.
Yeah,
thank you.
I will do.
And I also have a question. I don't see anything about the biophysics practical, whether on Moodle nor on Zoom.
So you are already inscribed in that course?
Yeah.
And via Moodle, you do not see, you mean this Biophysics Practical 2 course, right?
No, I think it's the #1.
Yeah.
It's a different, okay.
That's something different, yeah?
Okay, thank you.
Biophysics Practical course 2 is in the bachelor, the other is in the master, yeah?
Okay, thank you.
Okay, clarified, okay. No more questions? Bye-bye then, in 1 week again.
Bye.